Hello and welcome to this webinar on the generation of high-affinity recombinant antibodies for application in Immuno-MRM. I'm Amanda Turner and I'm the Product Manager for Custom Antibody Products at ABD Serotek. In a moment I'll introduce our speaker. First, I'd like to let you know that this webinar will be recorded and an on-demand version will be available shortly after the webinar on the ABD Serotek website. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up the technical guide which describes solutions for the most common technical problems. We'd like to make this webinar as interactive as possible, so please submit questions or comments via the Q&A box located at the bottom of the screen throughout the webinar. We'll answer as many questions as possible at the end of the presentation. Should any questions remain unanswered, we'll email you a reply. And now I'd like to introduce Dr. Christian Frisch. Christian is R&D Manager at ABD Serotek and has been head of the Antibody Generation Group since 2003. He studied molecular biology in Göttingen, Germany, and did his PhD with Professor Alan Furst at the University of Cambridge in the UK, working on the energetics of protein-protein interactions and protein folding. In 1997, he joined MorphoSys, working on the development of display technologies and the generation of antibody phage display libraries. In 2003, Christian became group leader at MorphoSys' new Research and Diagnostics Antibody division, Antibodies by Design, which was renamed as ABD Serotek in 2006, and in 2013 became part of Bio-Rad Laboratories. Christian's group at ABD Serotek has generated more than 20,000 antibodies in over 1,000 custom projects over the last 10 years. And now, over to you, Christian.
Thanks, Amanda. Multiple reaction monitoring mass spectrometry, or short MRM-MS, is an emergent technology for the quantitative analysis of peptide and protein expression with high precision and specificity. Such analysis of protein targets of interest has multiple applications. For example, in discovery research to look at changing levels of proteins upon disruption of a protein network, or in clinical research to detect proteins in plasma and tissue to verify and monitor human disease biomarker candidates. In my presentation, I will summarize a feasibility study to discover whether recombinant anti-peptide antibodies generated using the HUGO antibody library and phage display technology are suitable for immunoaffinity enrichment of peptides coupled to targeted MRM. Prior to performing MRM, many peptide analytes require an enrichment step to achieve sufficient sensitivity in the assay. Coupling a peptide immunoaffinity enrichment step to MRM, so performing an immuno-MRM assay, is effective for most analytes and provides high sensitivity and specificity. A prerequisite for performing the immuno-MRM assay is the availability of high-affinity antibodies binding to linear peptide in solution. The great expense and amount of time required for generation of hybridoma-based monoclonal antibodies has so far hindered the generation of immuno-MRM assays on a large scale. High-affinity recombinant antibody fragments like Fabs, isolated from large naive libraries, may offer several advantages over both polyclonal and monoclonal antibodies. The in vitro generation of Fabs in E. coli allows the user to control selection parameters, including affinity, to access a wide range of antigens, including toxic, conserved, or self-antigens, to automate the process and to complete the procedure more rapidly. However, no previous studies have investigated whether high-affinity recombinant antibodies against short linear peptides can be selected from a naive library, or whether the recombinant Fabs are suitable for the procedures involving immunocapture of small linear peptides. We therefore performed the pilot study together with a group of Amanda Pawlowicz at the Fred Hutchinson Cancer Research Center. 3 peptides were selected and antibodies generated against these peptides using HUGO and rapid maturation technology. The MRM-MS work with the selected antibodies was done at the FHCRC. Before going into the details of the pilot study, I would like to briefly introduce you to the immuno-MRM and to the HUGO technology. In preparation for immuno-MRM, a protein sample is digested into peptides, for example by trypsin, and one or several of these peptides are enriched by capture with an antibody coupled to magnetic beads. After washing, peptides are eluted and detected by mass spec. The measurement has a high precision, a linear range of greater than 4 logs, and absolute specificity. Proteotypic peptides are used as stoichiometric surrogates of proteins to quantify these proteins in biological samples. ImmunoMRM is reproducible across laboratories and capable of multiplexing several analytes simultaneously. MRM actually has been selected as Method of the Year in 2012 by Nature Methods. HUGO antibody technology is a unique and innovative method for the in vitro generation of highly specific, fully human monoclonal antibodies. It is based upon 2 important cornerstones: the Human Combinatorial Antibody Library, or short HUGO, and our advanced cis-display method of phage display. The HUGO Platinum phage display library contains more than 45 billion functional human antibody specificities, already prefabricated in an engineered Fab format. It has been optimized to increase the diversity of selected antibodies and the expression yield of candidate antibodies. In the HUGO Platinum library, the structural diversity of the human antibody repertoire is represented by 7 heavy chain and 6 light chain variable region genes, which give rise to 42 frameworks in the master library. These were combined with highly diversified complementarity determining regions, so the CDRs, synthesized using trinucleotide or TRIM technology to create a system that mimics the human antibody repertoire in vitro. Cis-display is a novel and proprietary phage display method that allows for the selection of high-affinity antibodies which are expressed on the surface of filamentous phage. It combines traditional monovalent phage display with a cleavable disulfide bond between the antibody and the phage. This allows efficient elution of all the captured phage particles in a manner that is independent of the antibody binding affinity to the antigen, which means that all phage displaying specific binders can be evaluated without a bias against those with high affinity. If necessary, the affinity of selected antibodies can be further optimized using pre-made trinucleotide CDR cassettes. A brief summary of the advantages of HUGO. It allows fast, high-throughput antibody generation. It's an in vitro technology, so selection can be guided to achieve high specificity requirements. Through the inbuilt optimization, assay sensitivity can be further enhanced if necessary. The library is synthetic and the selection is in vitro, and therefore antibodies can be generated also against toxic, conserved or self-antigens. The sequence of each antibody is known, so clone storage is infinite and the antibody genes can be manipulated or cloned into different formats. There are no HAMA effects since the antibodies are fully human, which makes them also the ideal source for calibrators and controls for in vitro diagnostic assays. Now back to the pilot study. Mouse osteopontin, human E-selectin, and human ADAM17 were selected as targets because they all had peptide antigens with high affinity monoclonal benchmark antibodies that had been generated previously by traditional immunization. The target peptide sequences are proteotypic for each of the respective proteins. For each antigen, 5 peptides were made. 2 peptides with artificial terminal cysteine for coupling to carrier proteins, one with a cysteine at the N-terminus and one with a cysteine at the C-terminus. Also 2 peptides with biotin at the N or C-terminus, and the free non-modified peptide for use in inhibition screening and for affinity measurements. This slide summarizes the phage display antibody selection strategy. Phage display selection, or panning, consists of antigen immobilization on magnetic beads, incubation with a HUGO library, removal of unspecific antibodies by wash steps on a Kingfisher instrument, followed by elution of the phage encoding the enriched antibodies. The eluted phage is amplified by infection of E. coli and production of new Fab displaying phage for the next panning round. 2 rounds of panning were performed on the peptide carrier protein conjugates coupled to magnetic epoxy beads using the respective transferrin peptide conjugate in the first and the BSA peptide conjugate in the second panning round. One panning was on the peptides with N-terminal cysteine, and in another panning the conjugates with the peptides with C-terminal cysteine were used. After the second panning round, the gene region coding for the CDR3s of the antibody light chains, so the LCDR3, from the pre-selected pool of Fab genes was exchanged with a highly diverse LCDR3 maturation cassette generated by trinucleotides, and E. coli cells were transformed with a ligated DNA. The obtained maturation libraries, which we call RepMat libraries for rapid maturation, contained between 2 and 9 times 10 to the 8 members. With a Fab displaying phage produced from these libraries, another 2 rounds of selection using increased stringency, so extended washing steps and reduced amount of antigen-coupled beads were performed. In one setting, the selection was again performed on the peptide carrier protein conjugates coupled to magnetic beads, using decreasing amounts of antigen-coupled beads in the panning rounds 3 and 4. In a second selection, the biotinylated peptides captured on streptavidin-coated magnetic beads were used. The change to biotinylated peptide in the RepMat panning rounds drove the selection towards the core peptide sequence and increased the number of antibodies binding the free peptide. After panning, the pool of Fab genes was subcloned into an E. coli expression vector, leading to functional periplasmic expression of monovalent Fabs, in this case equipped with V5 and strep peptide tags. The primary screening was done in ELISA on biotinylated peptide captured by Neutravidin. In addition, an inhibition screening was performed using free peptide as competitor. The clones with the highest signal in ELISA and showing the best inhibition by free peptide were sequenced, and all unique Fabs were produced and tested again in ELISA. The overall timeline for this process is 12 weeks. This is from panning start to purified and tested Fab antibody. Here is an example for the screening of anti-E-selectin peptide antibodies. 368 clones were screened in parallel on 2 plates. On plate 1, biotinylated peptide was captured by Neutravidin and then incubated with the Fabs in E. coli lysate from expression cultures. Detection was with anti-human Fab secondary antibody. Wells highlighted in green are ELISA signals greater than 10-fold over background, so there are many positives binding with good signal to the biotinylated peptide. To test whether the antibodies also bind the free peptide, a second screening was performed in parallel, where the Fabs were incubated with free peptide before incubation with captured biotinylated peptide. Most antibodies could be efficiently inhibited by free peptide. There are only few green signals on the screening plate 2. Clones with a high inhibition ratio shown below and high signal on biotinylated peptide were selected for sequencing. Several unique antibodies against E-selectin were identified, and the purified Fabs were tested again in ELISA. Control proteins BSA, CD33, and GST were coated, as well as the different E-selectin peptide conjugates, so peptide coupled to BSA and peptide coupled to human transferrin, and also the biotinylated peptide captured by Neutravidin. For the biotinylated peptide, again also an inhibition with free peptide was done. All antibodies were specific for the peptide and fully inhibited by the free peptide, so there is no dark blue bar showing up for any of the antibodies. This table summarizes the results of the screening for all 3 projects. For each peptide, a large number of clones was positive in ELISA and could be inhibited by free peptide. The antibody diversity was also very good. 9 unique antibodies were selected against the peptide from osteopontin from sequencing 40, 20 against E-selectin, we sequenced 42, and 17 against ADAM17, 33 clones were sequenced in this case. So really a very good diversity for antipeptide antibodies. For many of the antibodies, the affinity to the free peptide was measured, and the affinity ranges from high nanomolar to sub-nanomolar affinity. For each antigen, the 2 Fabs with the highest affinity for the free peptide and how they were selected are shown in this table. Both for osteopontin and E-selectin, antibodies with sub-nanomolar affinity were selected. affinity measurements was actually in solution using free peptide and monovalent Fab, and therefore the values represent the true affinity of the one-to-one interaction between antibody and peptide. 5 of the 6 Fabs and all the high-affinity antibodies were from the pannings using biotinylated peptide in the Reprint rounds. So this appears to be a good general strategy for selection of high-affinity antipeptide antibodies. All these 6 Fabs were converted to the human IgG1 format, and the IgGs were produced in mammalian cell culture. Further tests were done both with the antibodies in monovalent Fab format and in human IgG format. Just a reminder, if you not already have done so, please submit any questions you may have via the Q&A box at the bottom of the screen. Antibodies were tested in Immuno-MRM at the Fred Hutchinson Cancer Research Center. For immunoaffinity enrichment experiments, trypsin digested plasma, antipeptide antibody, a variable amount of heavy peptide, a constant amount of light peptide, and magnetic beads were mixed together in PBS containing 0.03% chaps. Synthetic light and heavy peptides were of greater than 95% purity. The heavy peptides were isotope labeled at the C-terminal arginine or lysine. For capture of the strep-tagged Fabs, strep-tactin magnetic beads were used, and capture of human IgGs as well as rabbit and mouse monoclonal antibodies was done with protein G magnetic beads. Peptides were eluted from the beads and analyzed by mass specs for quantitation. Initially, the performance of the HUGO and the traditional monoclonal antibodies was compared by estimating peptide recovery, which was determined by measuring the amount of spiked light peptide relative to heavy peptide prior to and following the enrichment process. 2 samples were prepared. In the first, a known amount of light synthetic peptide was measured relative to the stable isotope standard with no enrichment. In the second sample, the light peptide was added to diluted plasma digest and captured by the antipeptide antibody. Following the enrichment, the stable isotope standard was added and the ratio light-heavy measured by mass spectrometry. The relative ratio of light-heavy peptide in the 2 samples, so before and following the enrichment process, was used to estimate the recovery efficiency. As you can see in the last column of the table, the performance of our Fabs in this recovery experiments compares very well to the monoclonal antibodies. To further assess the performance characteristics of the antibodies in the Immuno-MRM assay format, we assessed the linear range, limit of detection, and precision for each combination of peptide antibody in a response curve in a complex plasma matrix. To eliminate any interference in signal from endogenous analyte, the amount of heavy stable isotope labeled synthetic peptide was varied, while keeping the light synthetic peptide at the same concentration in each sample. Response curves were constructed using the equivalent of 10 microliter of digested neat plasma as background matrix, and the enrichment step was performed in triplicate for each concentration point. The figure shows the response curve for the best anti-osteopontin peptide antibodies. Concentration ratio heavy light peptide is plotted against the ratio of measured peak areas for heavy and light peptide. The curves of Fab, human IgG1 and rabbit monoclonal are plotted together. The dynamic range of response is 4 orders of magnitude, and the curves for the different reagents overlay each other very well. The limit of detection for ABD 18303, both in Fab and IgG1 format, is even slightly better than the LOD of the rabbit antibody. So 0.3 nanogram per milliliter for the Fab and IgG from HUGO compared with 1 nanogram per milliliter for the rabbit antibody. Further investigation of the absolute peak areas shows higher signals for the osteopontin peptide using the ABD 18303 Fab and IgG. The light signal shown in red and heavy signal in blue are shown for each replica point of the response curve. The antibody type is denoted on the x-axis, so Fab, IgG, and MAP. Since the recovery efficiency for these antibodies was similar, the increase in peak area is likely due to a decrease in ion suppression from the background. So a decrease in non-specific binding and a correspondingly improved sensitivity of the HUGO Fab and IgG compared to the benchmark rabbit monoclonal antibody. The precisions expressed as % coefficient of variation for all three peptides are summarized in this table. %CV is based on 3 replicate captures of peptide and 10 microliter of plasma digest. Only points above the limit of detection are reported, and protein concentration in nanogram per milliliter is calculated assuming complete trypsin digestion. The precisions are comparable between Fab and the rabbit and mouse monoclonal antibodies. And although the best Fab for the ADAM17 peptide was only of medium, double-digit nanomolar affinity, it was still successful in immunocapture experiments and showed similar performance compared to the mouse monoclonal. This suggests that also antipeptide antibodies directly selected from the HUGO library could be used in the ImmunoMRM assays without employing affinity maturation steps, which could further shorten the timeline for Fab antibody generation from 12 to 8 weeks. To summarize, we could demonstrate in this pilot study that HUGO is a good source for high-affinity antipeptide antibodies. Against all 3 peptides, antibodies were generated that show excellent performance in ImmunoMRM. And for 2 of the 3 peptides, antibodies with sub-nanomolar affinity were obtained. We were also successful in employing streptactin beads to bind the Fabs through a strep tag, enabling the use of the Fabs directly in the assay. This saves additional preparation steps of converting the Fab to the full-length immunoglobulin. Results for the Fabs in the recovery experiments and the full response curve were comparable or superior to existing assays employing traditionally developed monoclonal antibodies. The use of the recombinant in vitro HUGO technology allows many projects to be performed in parallel and to complete projects in a relatively short time. A RAPMAT antibody generation takes 12 weeks and a standard antibody generation only 8 weeks, whereas generation of animal-derived monoclonal antibodies typically takes 6 to 9 months. I would like to finish by acknowledging the people who did the work. Jeff Whitaker and Lee Zhao from the lab of Amanda Pawlowicz at the Fred Hutchinson Cancer Research Center, and Stefan Hart, Francisco Hilera, and Achim Knappig at Bio-Rad ABD Serotec. The data presented in this talk have been published last year in the Journal of Proteome Research. If you want to learn more about our custom recombinant monoclonal antibody generation service or want to discuss a specific project, please contact our technical experts. With our established HUGO technology, we have generated over 20,000 antibodies in more than 1,000 projects since 2003, and our success rate is greater than 90%. And now I'm happy to take any questions you may have, and thank you for listening.
Thank you, Christian. As we wait for questions to come in, I'd like to remind everyone that the webinar will be available on demand on our website tomorrow, and everyone who registered will receive an email with a link to the recording. So we're just seeing if there's some questions coming in. Our first question is, are these anti-peptide antibodies that you mentioned in the presentation available via your catalogue?
Yeah, thanks. The antibodies are not in the catalog yet, but it is possible to get test samples. So please just contact us.
Okay, great, thanks. The next question is: have you used your method for generating antibodies for non-human proteins?
Yeah, I mean it's a synthetic library. And it can basically be used for any target. So not only human proteins, but proteins from any other species. And we have, we have a great variety of antigens that we generated antibodies against. So not only proteins, but peptides, as in the presentation, and haptens. We also pan on whole cells. So basically, you can any target you can think of can be used for this process.
Great, thanks, Christian. Our next question I will answer. The question is, could you please send a copy of the paper that you listed? Yes, we have an email that we can send out to you, which will have a link to the paper. We also have reprints of that. So for the person who asked the question, we'll contact you by email and ask for your address, and then we can actually send you a hard copy of the paper as well. And that accounts to the same answer to anyone who would like a copy of the paper as a reprint. Send us your details and we can put that in the post to you. It's also available as a short technical article on our website. We'll include the link to that in the email that we send you to answer that question. Okay, we have another question coming in, which is, have you compared your antipeptide antibodies to cis-kappa?
I actually probably we have to go back to this person again, because I mean this is basically just a different name for this mass spec technology. So basically this was tried in this technology. We probably have to get back by email later.
Okay, thanks. To the person who asked that question, we'll contact you and see what else we can help you with regarding that answer. We have another question come in, which is, what about conformation-dependent epitopes?
Yeah, I mean, conformation-dependent epitopes. It's best to use the native protein as a target, but also for peptides, there are technologies, and we've also used them for generation of antipeptide antibodies, where you fix your peptide, or where you fix a loop conformation by using two cysteines, for example. So there are several strategies to generate conformation-dependent antibodies, and also here it's best to contact us directly with a particular project in mind.
Great, thanks. That also answers another question, a very similar question that's come in, so I hope that answers the other question too. One more question about the antigens that we use is, could this technology be used for generation of antibodies to small molecules like opioids?
To smaller molecules is as I briefly mentioned, is also a target class that we have relatively often. The thing here is that these small molecules need to be somehow coupled, best coupled to carrier proteins, so that they can be presented to the antibody library. So it's not possible to just use the small molecule, because that cannot be presented to the library. But once you have this small molecule coupled, then you can, similar to this strategy that I've just presented, you have your coupled small molecule, and then you can either use the free small molecule in the panning process already for elution. And what we definitely always do is an inhibition screening with a free small molecule. So it's basically a very similar process to what I've just presented.
Great, thanks, Christian. We have another question about antibody production. That is, how easy is it to scale up antibody production?
To the 100 milligram scale. It's very easy. It's just using larger. I mean, go from smaller bottles to shake flask, or so producing in the 10 to 100 milligram scale with our E. coli system is absolutely no problem. Scaling up to larger than that, I mean, this is also something that you would need to to talk with us. We have a system running which we can use for gram scale expression.
Okay, thank you. The next question is application related. Have you tested these antibodies in other applications such as Western blotting or IHC?
We've not actually. This was maybe these antibodies, there's a possibility that these antibodies do not work in Western blot because we've left one terminus of the peptide free for detection of the antibodies, and in Western blot, the whole protein, this terminus would not be a terminus, but be an internal part of the protein. So if the epitope includes the N or C terminus of the peptide, then it may be that these antibodies will not work in Western blot, but that has not been tested so far.
Okay, thanks. Another question is, can you generate anti-idiotypic antibodies?
Oh yeah, that's a good one actually. We do have a webinar on our website about that. So this is a particular strength of HUGO, the generation of anti-idiotypic antibodies. So we've a number of anti-idiotypic antibodies in our catalog already, and we also do many custom projects for generation of anti-idiotypic antibodies. So please have a look at our website. There's a webinar and a lot of information.
And for the person who asked that question, we will email you with some links and some information that will answer a bit more for you in detail about our capabilities for anti-idiotypic antibodies. Okay, so for now, I think we've answered all the questions that have come in. I'm just checking to see if I've missed any. I think that's it, actually. So we've reached the end of our Q&A session. Thank you to everyone for listening, and we look forward to you joining us again for future webinars.